Key properties of neuronal cell membranes. This image introduces the key properties of neuronal cell membranes and explains how neurons generate electrical signals. The cell membrane separates the inside of the neuron from the extracellular fluid and creates an electrical difference known as the membrane potential. This membrane potential is essential for the formation and conduction of nerve impulses. The membrane contains specialized proteins called ion channels. These channels regulate the movement of ions across the membrane, allowing the neuron to communicate with other cells. There are two general categories of ion channels. Leakage channels, also called non-gated channels, are always open. They allow ions to move continuously across the membrane and play an important role in establishing the resting membrane potential. Gated channels open and close only when they receive a specific stimulus. These channels allow the neuron to rapidly change its membrane potential during nerve impulse generation. There are three major types of gated ion channels. First are chemically gated channels. These channels open when a chemical messenger, such as a neurotransmitter, hormone, or ion, binds to the channel. They are commonly found on dendrites in cell bodies, where they receive signals from other neurons. Second are voltage-gated channels. These channels respond to changes in membrane potential. They are primarily located along the axon and are responsible for generating and propagating action potentials. The two most important voltage-gated channels are sodium channels and potassium channels. Third are mechanically gated channels. These channels open when the membrane is physically deformed by pressure, stretch, vibration, or touch. They are commonly found in sensory receptors throughout the body. Let's take a closer look at the voltage-gated sodium channel. When a neuron is at rest, the sodium channel has two gates. The activation gate, located near the outside of the membrane, is closed. The inactivation gate, located near the inside of the membrane, is open. Because the activation gate is closed, sodium ions cannot enter the neuron. When the membrane reaches threshold, the activation gate opens rapidly. Sodium ions rush into the neuron, causing the membrane potential to become more positive. This process is called depolarization. Very shortly afterward, the inactivation gate swings closed, stopping the flow of sodium ions even though the activation gate remains open. This brief inactivation helps ensure that the action potential travels in only one direction. Next are the voltage-gated potassium channels. Unlike sodium channels, potassium channels have a single gate and respond more slowly to depolarization. As the sodium channels begin to inactivate, the potassium channels open. Potassium ions move out of the neuron, carrying positive charge with them. This outward movement of potassium returns the membrane potential toward its resting value, a process known as repolarization. Because potassium channels close slowly, potassium continues leaving the cell for a short time after the resting membrane potential has been reached. This causes a temporary hyperpolarization in which the membrane becomes slightly more negative than its normal resting value. The potassium channels eventually close, and the sodium-potassium pump, together with the leakage channels, restores and maintains the normal ion distribution across the membrane. Together, leakage channels, gated ion channels, and the sodium-potassium pump allow neurons to generate rapid electrical signals. These specialized membrane properties make it possible for the nervous system to receive information, process it, and communicate with muscles, glands, and other neurons throughout the body.